Just want to welcome everybody to the United Leukodystrophy Foundation's virtual family conference session on Alexander disease, presented by Dr. Messing, Dr. Waldman, and then from Ionis, Jordan McSherry and Ann Smith. And I will turn it over now to Dr. Messing. Thank you.
Thank you, Tara. Let me start sharing my screen right now and tell me if you can see the first slide.
Perfect.
Is that visible?
Looks good.
Okay, great. So, so thank you to the ULF for hosting this meeting and giving us this session on Alexander disease. There's certainly lots of interest in what's going on, and I will move right into it. So I'll accept that my keynote doesn't seem to advance in this mode. So I think I'll just go back to, I'm gonna have to stop sharing and then start it again. Okay, you can see it now?
Yes.
Okay. So first of all, I'm going to take 30 seconds and do a bit of recruiting. There may be some developmental pediatricians who are listening in now or will listen later on. If you have any trainees who are ready for their own positions, the Waisman Center has a position open. And if you're interested in that, please contact Chung Chang, the director of the Waisman Center, at qchang@waisman.wisc.edu. So the way we've organized today's session, it's going to be the first part, which is the smallest part, on our stuff involving preclinical models, and then the second portion, which will be we've allocated much more time to, be updates on the clinical trial from Ionis and CHOP, represented by Ann Smith and Amy Waldman. So let me start with our things. Just a few quick things. Many of you may or may not know that several years ago, I guess in 2018, I wrote a short book entitled Alexander Disease, A Guide for Patients and Families. A lot's happened since 2018. The bad news is that the publisher is no longer interested in really selling it. And so the good news is that I decided to provide my own update to it, and it'll be free. So hopefully sometime in July, I'll have that organized, and I'll send out the links for where you could get it to the distribution list. And by the way, if anyone wants to get that beyond that distribution list, you should just send me an email with your name and email address. Another new thing that we're going to start, hopefully in July, will be podcasts offering some discussion about one or more of the recent publications. I've been typically sending out updates once a month that just list the publications that have come out in the past month. And one of the comments that's been made about that is that while people really appreciate getting the list, they often would like some idea of what the significance of it is. So, so the first one will be sometime in July. I guess at this point it'll be later in July, and my first guest will be Michael Brenner from University of Alabama, Birmingham, with whom I've collaborated a long time. And I'll send out the information about where to get that podcast as well to the distribution list. So let me turn attention now to this. The main topic I was going to present is this new rat model that we've developed for Alexander disease, and why we think this is such a major advance for the field. Some of you, we have talked about some aspects of it in public before. Those of you who were at the 2018 conference in Madison heard heard something about it. Some of it was presented at the 2019 conference at ELA. But we've held off really trying to publish it or submitting it for publication because we felt we needed to nail down one really important aspect, which is whether it has a leukodystrophy, and it turns out that it does. So, in general, over the years, we and others have developed several models of Alexander disease involving cell culture and fruit flies. Certainly the mouse models have been around for a long time. Recently, some zebrafish models have been made. So the question is, why bother making a rat model? And there are several potential advantages of a rat model that made us interested in trying it. One is that there have already been several examples where rats more closely resemble the human disease counterpart compared to the mouse models. They're certainly more amenable to behavioral assessment than mice are, especially including cognitive testing. There's more tissue and body fluids available for analysis just because they're bigger. And we're very interested in using things like blood and cerebrospinal fluid to try to track progression or response to treatment. And hopefully there might be more improved clinical measures for testing the effects of new therapy. So there was lots of reasons why we wanted to pursue making a rat model. As many of you know, there are over 100 different variants in GFAP that have been associated with Alexander disease, and it's just shown diagrammatically on the right-hand portion of this slide. What we decided to do was to engineer one of the more common and consistently severe variants in human, R239H, which in the rat is numbered R237. So basically, we engineered that as the same type of point mutation in the rat genome that would produce this single amino acid change and produce animals that are heterozygous for that R237H variant. One of the first striking things about these animals is that while they're born in normal numbers and they seem to be fine within the first couple of weeks of life, things really change dramatically at about weaning, which is for a rat is at 3 weeks. And on the left-hand side, you see a bird's eye view of a cage of 2 litter mates. These are 8-week-old females. This is the wild type female here and her litter mate that carries the R237H variant over here. And tracking this in terms of body weight on the right-hand side of the slide, this is the curve for the growth curve for wild type rats, and here's the growth curve for the R237H rats. So they basically stop growing for a several week period of time. Eventually, they start to put on weight, but never really achieve the weights of their wild type litter mates. Now, another thing that was never really, and I'm trying to stress the things that are different about the rats than the mice that make them such an important advance. Another thing that never really happened with the mice was mortality. And they essentially, the point mutant mice, that they're essentially the same variant as this, have a normal lifespan. In the rats, we do have increased mortality in this period of 8 to 12 weeks. So beginning right about here, you see a loss of about 14% of the animals. And then after 12 weeks, if they make it past 12 weeks, then they have a normal lifespan. And so we don't really know exactly why they're dying. This small subset is dying at this point, but they do show that measurable mortality. Another thing that's very different is that the rats, unlike the mice, have significant motor deficits. So now we're moving into the arena of what clinical clinically relevant symptoms do they have? And we have various ways of testing that in rats, but one is what's called a horizontal ladder. It's kind of like a jungle gym that you hang from with your hands on the playground, but for the rats, they're crawling across it from the top. And on this slide, we've got, so here's the horizontal ladder here, and we've got a wild type rat. I've got to turn off the sound. Just cruising along, and an 8-week-old R237H litter mate, making it across, but requiring regular stops, and has multiple episodes of slipping, where his or her foot slips through the bars. So, and you can quantify these kinds of slips. So, so they certainly have deficits in motor function. It's really interesting that if you just look at spinal cord diameter, and now we're looking at a cross section of the cervical spinal cord, a wild type rat on the left, and one of the mutant rats on the right, there is definite atrophy, or the mutant spinal cord is definitely smaller than the wild type spinal cord. And this is somewhat reminiscent of what we see in especially the type 2 Alexander patients in human. And if you measure the different areas of the spinal cord, this is the total area. You can see the mutants are much smaller than the wild type. Notice the y-axis does not go down to 0 in each case. So because we're just trying to highlight the differences here. But the total area is really different, and it's really because there's a change in the white matter. The gray matter is essentially the same size. So we're very excited about this because, of course, Alexander disease is a leukodystrophy. We've never had an animal model before that exhibited any evidence of a leukodystrophy or change in myelin. So we pursued additional studies to really focus in on myelin. And this slide shows quantitation of myelin proteins by Western blot, shown on the left-hand side, myelin basic protein. There are 3 different sizes of it, as well as 2 others, PLP and CNP. And basically all 3 of the myelin basic protein isoforms are reduced in the mutants compared to the wild type. PLP is essentially unchanged, and CNP shows a very, very modest change. It's statistically significant, but it's not nearly as dramatic as the changes in myelin basic protein. And if you look at the myelin morphologically, in this case, by electron microscopy, here we're looking on the left-hand side at a region of the spinal cord, the dorsal corticospinal tract, the wild type animal over here on the far left, and right next to it at the same magnification, the dorsal corticospinal tract from one of the mutant animals. And even at this fairly low magnification, you can appreciate that there is a change in the myelin. It's not completely absent like you might see in other disorders of myelin, but the myelin seems to be thinner, and there are many areas that may even be devoid of myelin. If you look at this same tract in the mutants at higher magnification, you could also see several axons that maybe that have certainly thinner myelin sheaths, and also may be undergoing degeneration with ballooning of these intracellular structures, or complete condensation into these dark masses like shown on the right-hand side. So there's clearly a significant change in the myelinated fiber tracts in these animals. So the last slide I'll show then is, of course, are they responsive to antisense oligonucleotide treatment. And what we did, what we published in 2018 was the first study in the mouse. And fortunately, those antisense oligos also recognized rat GFAP. So here we've got a video showing 2 mutants and 1 wild type. And of the mutants, one of them is treated with antisense, and one of them is treated with phosphate buffered saline or vehicle. And you're really be hard pressed to tell which one is which in this, because basically one of these 2 rats is an R237H rat, and it turns out to be this one treated with ASO. So, you know, I think it was the mouse data and the rat data that provided a lot of the motivation to go forward with a program to develop antisense as a clinical therapeutic and something that was worthy of testing in a clinical trial in humans. So the summary is that we think the rats are a major new resource for current and future research. They exhibit all the molecular features that we expect, plus abundant Rosenthal fibers, leukodystrophy, impaired motor function, reduced body weight, and some mortality. And an early single treatment with antisense prevents nearly all of the phenotype. It lasts at least 24 weeks. And treating late at the sort of the peak period of disease, achieves at least some reversal of the phenotype. So with that, let me just thank at least 2 of the people who were really key in driving this forward. Tracy Hageman from the Waisman Center and Barrett Powers from Ionis. I think with that, of course, I'll be happy to answer questions, but maybe now I should stop sharing and turn it over to Amy and Anne, and then there's going to be an open-ended question and answer session at the end.
I'll be real quick. There was a question in the chat. Can you share your email address again?
Yes, this is, let me just put it up there then.
Perfect.
Okay, is that, it would be on this slide. amessing@wisc.edu. Okay.
Okay. Great. Well, thank you, Albie. It's always humbling to speak after you. I think I speak for our entire community in thanking you, Barrett, Ionis, for everything that you have done for our Alexander disease community. And I actually was not going to be here today, but couldn't resist the opportunity to talk to everyone and say hello and update everyone. So on behalf of Ann Smith, who is here as well, and some other representatives from Ionis, I wanted to talk to you a little bit about the clinical trial. So I am wearing a different hat when I'm talking about the clinical trial. Our natural history and our research studies at CHOP are still ongoing, but now I'm putting on a different hat in talking about the clinical trial, and happy to review some of the things that we have said a few months ago and take some questions. So just to get started, so literally getting started. So we did want to share with everyone that the study has begun. So the clinical trial has officially started. It has been launched officially at 2 sites, and we'll go through that in a few minutes, with some other sites coming on board shortly. So the study has begun. And just as a review, I think many people know, but I think it's important just to reiterate that this is a clinical trial. So this is a research study. This is not clinical care, where study participants either receive the study drug, which is called Ion 373, or placebo. And so the study is, you have a spinal tap performed every 12 weeks. So every 3 months, patients undergo a spinal tap, and they will either get placebo through that spinal tap or drug. The placebo group will have placebo for the first 5 doses, and then at 15 months or after 5 doses, will receive drug. So eventually, everyone does receive drug. But some 2/3 of the patients will be, in general, will be randomized to drug, and about 1/3 will be randomized to placebo. None of us actually know whether the patient is going to get the drug or a placebo. That's the idea behind a randomized controlled trial. So I don't know, you don't know, Ionis doesn't know, and I would encourage you not to try to guess. I know that that's hard, and everyone wants to kind of look, but really the idea here is to let science and this method, the scientific method, kind of give us the answer. So we really just want to know, how is your loved one doing? Not necessarily, do you think you're getting placebo or drug? Okay, so there we go. So I do want to get into a little bit more detail. We've gotten a lot of questions about the information that's on clinicaltrials.gov. And while there is a broad age range, 2 to 65 for this study, that does not mean that everyone who's 2 to 65 is necessarily eligible for the trial or going to be treated at the same time. So there are some ways that the trial was built and designed that there will be certain age groups that come on board perhaps before other age groups. And there may be a lot of people in this age range that actually don't ultimately meet eligibility criteria. So there's a little bit more information than just everyone 2 to 65 can be in this trial. So what happens during the trial, not only do you come for the treatment, the spinal tap, but there are a lot of tests and questionnaires to measure whether the drug is working. So a clinical trial is all about measurement to say, does a drug benefit the patient? As everyone I think has heard a little bit, there are going to be several clinical sites worldwide. That is the goal, that is the hope. We are trying to get, and Ionis is trying to negotiate with many of the sites throughout the world. And once a site in a country is named, then the cities that are part of the study that are hoping to be part of the trial will also be named on clinicaltrials.gov. And as I mentioned, the primary goal of the study is to assess the safety and tolerability, and just as important, measure changes in motor function, communication, cognition, and everyday functioning. So it's really the design of a clinical trial is to show that there is some benefit, and of course also show safety. Just to, I don't know what happened there, sorry. And should I keep going, or are there questions on this slide that we should answer? I can't see the questions while we're presenting.
I think you can keep going. There is a question about repeated dosing, but I'm going to answer it in the chat.
Okay, perfect. Okay, so I just want to walk everyone through what the clinical trial participation looks like. So everything begins with informed consent. And this is where we describe the study in detail, your obligations if enrolled. And just to pause there for a second, this is really your promise to us and the clinical trial site that you are going to prioritize your participation in this trial, right? So that the idea is that you're committing to this trial, whether or not you receive placebo or not, with the intent to stick through the entire trial, right? So knowing that you're going to have to go to the site every 3 months, you're going to have to go through additional evaluations in between those 3 months. So really that these are our ask of you if you participate in the trial. Now, this is a research subject and a research trial, and you do have rights. And as you can see on the bottom of this column, you also could withdraw from the trial. And you're not obligated, even though we use that word, you're not obligated to fulfill it, but our hope is that you really do come into the trial with the intent to stick it through, of course, unless there's a reason not to do so. And I want people to think about that it is a several-year commitment to participating in this trial. If someone does drop out, which does sometimes happens, that contributes to the overall, that causes some delay sometimes in it being able to analyze the data and then fulfilling the number of patients. So really the consent process is really understanding what you're committing to and then your rights as a research subject. So this is a time where you should really review this document in detail, understand the implications, understand the risk of participating in research. And so this is a time for you to ask questions and to really go through this document very carefully with the study team. And then if you decide to participate, you sign the consent form. For children participating in the study that are under 18, if possible, they should also assent or give their permission. So there's a process that involves a child agreeing to participate. Not all children can verbalize that, and that's okay. And then, as I mentioned, you can withdraw and leave the study, but we would ask that that would be for a medical purpose and not necessarily because you just can't commit to the study anymore. So I really want to emphasize that this is a several-year commitment. Once the consent process has occurred, then we start the screening process. And this is an in-person evaluation at the study site to determine eligibility. So even if you have all those check boxes or you meet all of the criteria that that have been discussed or are listed on clinicaltrials.gov, there are still additional screening criteria. So not everyone that screens is necessarily automatically eligible. Our hope is that many people will be eligible that screen, but there still are some additional steps that we need to make sure that you meet additional criteria. There's some blood work, there's some actually physical motor tests. So just to reiterate, our hope is that everyone who screens is eligible, but the investigators don't necessarily know that at the time that they screen individual patients. You have to go through the screening and then we can determine eligibility. So this involves a couple of days where you do functional tests and questionnaires, blood tests, but no lumbar puncture as part of the screening. So the visit often spans, 2 or 3 days. And then after that, we take all of the information to determine eligibility. Okay, so, and just one last point there, you sign the consent form in order to be screened, and then you're screened to get the go-ahead that you're eligible. All right, and then patients that are eligible can move forward, enter this double-blind phase, as we talked about. This involves several in-person visits to the study site. So there's lots of visits. There are visits in between the lumbar punctures. There's also going to be some phone calls to check in. So there's a lot of work on your part. And the assessments are going to be similar to the screening, except that there are a few MRIs throughout the study. Not at every visit, but there are some key MRI points as well. There's the lumbar puncture every 3 months, additional assessments if necessary for individual health. And as we talked about, the double-blind phase of the study will be 5 doses of drug or 5 doses of placebo. After that, we get to the open label phase, and this is where everyone gets drug. So the assessments are the same. It's still a spinal tap every 3 months, but everyone will get drug. And the assessments, the motor tests, the questionnaires are all of the same. And there's 5 doses of drug during the open label phase. All right, any questions on the clinical trial participation? Okay, I just wanted to let everyone know where they can find updates. So clinicaltrials.gov is really the bread and butter of all of the updates and the official updates for the trial. You can either type in Alexander disease where it says condition or disease, or type Ion 373, whichever you remember. And then what you'll see is one study found, or sometimes for Alexander disease, a couple studies are found, but you're looking for the one that talks about a clinical trial of Ion 373. And if you click on that, it will again list the key inclusion criteria and then locations. Again, locations only gets updated once a site in a country is added, but will not add sites at other countries until at least one site in that country is active. So we have one site that's active in Canada, which is in McGill in Montreal, and we have CHOP, which is active in the United States, and Stanford, and then other sites will be added as they become active. But because we have sites in those 2 countries, those are the countries that are leading, and Ionis is working on contracts and other things with other sites throughout the world, around the world. But this is the site that's going to be most updated when a site becomes active. And then there's also some other resources. So Ionis is a website that lists all the trials that are going, that Ionis is hosting, not just Ion 373. So you just have to scroll through that site to look for this. There's a place if you need information that you can find. So it is a very user-friendly site with information about the trial. And then there's lots of community partners that we can distribute information through as well. And then conferences such as this one. So grateful to the ULF for allowing us to use some of the time from this family meeting to talk today. You know, and while I'm sort of stopping sharing and trying to look at the chat and answer some questions, you know, I do want to also mention that, you know, we are here obviously speaking about the trial, but we do recognize children and adults who are not eligible for the trial or not able to participate in the trial. And all of our sites, whether it's a clinical trial site or just a leukodystrophy expert and physician, are here and committed to the well-being of all of our Alexander disease patients. And your care is just as important to us as the clinical trial. So we can continue to use this time to talk about the clinical trial or anything else that you might have questions about. All right, I'm going to stop sharing just so we can see the chat.
Amy, it's Anne. I can give you one from the chat that I didn't answer. I thought you would want to take it.
Okay.
There was a question, has anything changed with the Philadelphia site not allowing under 18s to participate at this time?
Yep. Thank you for bringing that up. Okay, so just to reiterate, let me just explain to the community where that's coming from. So as you see, the clinical trial is available for ages 2 to 65. And the CHOP Institutional Review Board, actually all institutional review boards, are really mandated to minimize risks in children, right? So the goal is, when you're bringing a new drug to children, is minimize risk. I think we can all appreciate what happened with the COVID vaccine, where adults needed to be vaccinated first, show that it was safe before it could be given to children. The CHOP Institutional Review Board said the same thing. Well, if you can give this to adults because adults are eligible, we want you to give it to adults before you can give it to children. Now, we wrote back and said, but Alexander disease is different in different age groups, and we should be able to avail these children of this potential, life-changing therapy. Again, it is a clinical trial, so we don't know for sure that the drug will be effective. We have the animal data to say that it was effective in animals, but we still don't have proof that it works in humans because it's a new drug. So the CHOP Institutional Review Board said, we appreciate that children are affected differently, but we want to see that it's safe first in adults. So we do have a plan and a time frame of when we think we're going to go back to the Institutional Review Board at CHOP and present data on a certain number of adults so that we could then allow children into the trial at CHOP. I just can't make those guarantees or promises. We're hopeful, we're optimistic that with some data on adults that they will change their mind and say, yes, now it's safe for you to allow children. I can't be 100% certain, but I think all of the patients that know me on this call know that I will not give up until we've tried everything we can to show that the drug is safe enough to be given to children. So that's how the CHOP Institutional Review Board approached the study. I will say that other institutional review boards had similar questions. They just came to a different conclusion, and many are more comfortable allowing children with the design that Ionis has proposed, which does start with some older patients and then before we're able to give it to younger. So other institutional review boards were a little bit more lenient, I guess is the right word, in terms of how this trial was designed, because Ionis did design a trial to minimize risk in kids. And so we're hopeful, I almost flipped there. We're hopeful at a certain period of time that we might be able to enroll kids, but for right now, we're only enrolling adults.
All right. So Tara asked that I read the responses I typed. I think I did not do this right. So let me read the few that I typed here, and then Amy, that'll give you a chance to take a look at some more that have come in. So we got a question about the rat model, and if a single dose was successful in the rat model, why do we need to do multiple doses in humans, which is an excellent question. And the answer is that the ASO is not permanent. So I think some of you have heard me talk about the central dogma of molecular biology before, where DNA makes RNA, makes proteins, and a change to the DNA would be permanent, but that is not how ASOs work. We work on the RNA. So we're trying to intercept that message that is ultimately made into a protein. And by intercepting the message, we prevent the protein from being made. So the RNA is going to continue to be made while an ASO is given. And if the ASO is present, it can intercept the RNA and lower the amount of protein that's made. But the ASO is going to dissipate over time. It's like any other drug. Like if you take an Advil, it's going to be in your system for a while and then it's going to go away. So ASOs are, if they're effective, they can be treatments for diseases, but they cannot be cures for diseases. We're not a gene therapy. But one of the things we'll learn in this trial is how frequently we need to give the drug. Right now, we think every 12 weeks is probably a good regimen. But we'll take measurements during this study and learn whether that's too often or it could be stretched out further, so that we can have that information going forward to best serve the community. And then I answered another one. Oh, there was a question about where is the LP, and it's down low. So you have your spinal column and the spinal cord extends down through it. We're going in below where that spinal cord ends into the open space where it's just CSF. I put in the chat that usually L3, L4 is the location because somebody asked for a specific location, but whoever's doing the LP is going to sort of feel around there and pick the best spot for you, but it's down in that low area. All right, Amy, do you want to tackle some of the others that came in?
Yeah, sure. Okay, so if a patient is under 18 and ineligible, I think that's mostly at the CHOP site, because there are some other sites that are taking under 18, and then opens to under 18, but the patient then ages out and is 18, yes, they would still be considered. CHOP, my goal or my hope, even though CHOP is now taking 18 and above, that when it opens up, it would be really reaching the full 2 to 65 criteria that was set out by Ionis in terms of the trial. I will just mention one other thing. I wrote a note to myself earlier to mention this. You know, in terms of being eligible for this trial, this isn't something that, you know, that I chose or Ann chose or any of us sort of, we didn't choose eligibility criteria. This is really statistically driven. That's really how this works, is that there was a lot of data that came from many of you through participation in the natural history study that went into these algorithms and statistical models. And that's really how a clinical trial is designed, really through statistics and stuff. It's not so much choosing or picking people or criteria, because again, the goal of a clinical trial is to show a measurable change, right? So it's all about numbers and statistics. So I just wanted to mention that. In terms of the other sites, so again, the trial is 2 to 65. It is a little complicated in that some of the sites might be more children's hospitals, some of them might be more adult hospitals. Like, it is actually kind of ironic that CHOP as a children's hospital is doing adults, but luckily we're able to see adults, and we're able to have adults stay overnight at a children's hospital for monitoring after the first dose, which is part of the protocol. So it's not necessarily clear, unless, and if I'm saying something wrong, please jump in, that every single site is going to be able to take children and adults from 2 to 65. I think we are doing our best, and Ionis is doing the best to try to anticipate what sites will be able to take adults and what sites might not, and vice versa with children. It's still a little bit early to predict every site in terms of what they'll be able to do, but Ionis's website is trying to actually fill in as much as they can in terms of whether they'll be starting with adults or starting with children.
Yeah, I'm looking, actually, we intend, yeah, so clinicaltrials.gov right now. Amy showed you how to get there. Sorry, Dr. Waldman showed you how to get there. When sites are listed, if there is a children adult breakdown at that site, it's going to be shown on clinicaltrials.gov. So I'm looking at it right now, and it's there right now, and it says Children's Hospital of Philadelphia currently recruiting adults only. The Chicago site will initially recruiting children only. Sorry, we need to fix our grammar, but basically that information per site, if there is some sort of restriction in one direction or the other, it's going to be shown to you on clinicaltrials.gov.
A few more things. So who shall initiate the contact for interest for the trial, parents or clinicians in charge? Either. So, a clinician, and actually sometimes both, a clinician can certainly refer us, but for a research study, the way this works is we actually cannot solicit people to participate in the trial. It is actually best if you contact us to say that you're interested in the trial, and then we can open the dialogue. There are just some rules about that. So even if a physician emails me or a clinician emails me, I really need to hear from the patient to say, I am interested in the trial. So on clinicaltrials.gov, it does list the contact at each site. And so I would just encourage patients and their families. Well, it actually has to be the patient themselves if over 18. It can't be the mom or anybody. It has to be the patient if over 18 that contacts the site. Of course, if it's a child under 18, then it could be the parent at the sites that are able to take children. So really best for you to reach out. Your clinician can as well. The other thing that I will mention is there are really coordinated efforts to bring patients to the sites and to enroll in the trial study-wide. So as much as we can, we're in touch with the monitors of the study, and the monitors then kind of coordinate between the sites. So sometimes there's a pause or a wait. We all thought that this was going to be rush out of the gate, that we were all going to enroll patients into this trial and dose. It's actually a slower process. Dr. Smith tried to warn me about that, but I didn't really fully appreciate it. So I would encourage you to stay in touch with the site because things are changing week to week or month to month. And so if you are interested, send an email. If you don't hear anything for a month or so, that's probably because there's a lot of coordinated efforts going on. Please resend that email. Let us know, keep in touch, let us know that you're interested. There are some planned sites in Europe, and I'm hopeful in the UK. I think we're going to have to wait until a lot more of those contracts, and I don't know if it's laws or other things, are further along in order to share more information about that. But I do give Ionis a ton of credit. They have really tried to plan sites around the world, but there are different, I don't know, Anna, is it considered it's different laws or different rules or regulations when sites can onboard?
It's just a lot of paperwork.
Yeah.
A lot of paperwork, a lot of meetings, a lot of questions and answers. Just to share, it can take, I don't know, the fastest site I've ever seen get open might be 4 months, but it can take well over a year in some countries, at some institutions, just because there are a lot of hurdles to getting there. So we have a lot of sites in a lot of countries engaged and interested and doing the work to get started. And as soon as we have the country and the local approval for a site in a country, you'll see that country pop up on clinicaltrials.gov.
Yeah, and with CHOP being very aggressive and me hounding people very frequently, it still took us 10 months. From the time that we sort of initiated the contracts to the time we were able to really get going. It's and after that, it really does actually take another few months to really, once contracts are signed, then there's a lot of motion that happens actually at the site to then execute the trial. So the contracting part and everything takes a long time. And we really put it into high gear, and it still took a very long time. So I thought there was a great question. Due to all the different mutations, is it possible that different patients would ultimately need different dosing schedules? It's a great question. It's we are looking at, even though they're different mutations, there's the common factor that everyone has elevated GFAP. So right now we're thinking that it's going to be a similar dosing and similar frequency once we know more about the drug, but that's the purpose of doing all of these serial measures over time, so that we can see what happens to the GFAP levels and how quickly they drop. And that is a great question. I would say we are working on a paper that hopefully Albie can showcase soon this summer, where we have looked at different mutations and different GFAP levels. And I can say that I can't tell right now, based on the mutation, what someone's GFAP level is going to be. Maybe age plays a little bit of a role, but thanks to many of you that are participating here and giving your blood and your spinal fluid, we should have some more answers to that later this summer.
Anne, do you want to comment on this question about Alexander disease and Huntington's disease?
Yeah, I was just scrolling back up. I think we missed a few. We missed, if this works, would the treatment, the lumbar puncture, be lifelong? And I hope we've answered that question. This ASOs as a class are treatments. So for as long as you want the drug to knock down the amount of GFAP, as long as that is beneficial, you would need to keep taking the drug. Let's see. If the treatment works, can the participants continue with the treatment after the end of the trial? That's something that we don't know today. We work our way through this first experiment and we learn what we can about the drug. Is it safe? Do patients benefit? It's not unusual if a drug looks promising in a first trial that a company will find a way to make sure that those heroes who sign up for that very first trial continue through another protocol, maybe an open label extension. There are a number of different mechanisms to have a drug available if the company is still developing it. But right now it's premature. We need to learn here if the drug is safe and if patients benefit. Then the Huntington question. Do we know if Alexander and Huntington's disease act similarly? So what we know that's similar is that in Huntington's disease, there is a protein called the Huntington protein that is toxic. In Alexander disease, we know that the GFAP protein is toxic. So there's that similarity, which is what attracted Ionis as a company to try an ASO in both of these situations. But this question, in case anybody's not aware, probably comes from Roche, who we are partnered with on Huntington's. Used an ASO in a phase 3 trial, and they recently stopped that trial because a monitoring board that helps them with decision making suggested it was unlikely that the trial would be successful in the end. It was unlikely that the drug was benefiting patients. We're working with Roche now to understand the data that came from that trial. There are a lot of ways for trials to fail. We can fail drugs and drugs can fail us. And we don't yet know if the Huntington ASO is a good drug or not. Some ways that we fail drugs is doing the wrong trial, not properly designing a trial, either the patient population, the inclusion-exclusion criteria, the endpoints that you choose, the duration, the dose, the regimen. There are a lot of decisions that are made based on our best judgment. But sometimes a trial doesn't give a drug the best chance to succeed. So we don't know if that's the situation yet with Huntington. What we do know, the little bit that we do know so far, doesn't feel like an impact that's going to translate to other programs. There's nothing that we see in the safety or the pattern in that Huntington trial that would suggest it's going to flow through to anything else. But that's something that we'll look at over the next, I don't know, it's going to take 6 or 12 months to sift through those Huntington data and really understand the situation. There are a lot of differences between the disease. Huntington as a protein is a protein that's essential in early development, and its role later in life is relatively unknown. So there's a bit more risk with knocking down the Huntington protein than GFAP. Here we know from Albie's work and others that GFAP can be completely knocked out and not have an impact. So we have a bit more of a safety margin here as well. I'm sort of rambling there, so I'll stop. If there are follow-up questions on that, let me know. But the big picture is we're learning from the Huntington program, and at this stage, we don't expect any follow-through to other programs.
Great. Okay, so Dr. Smith, I'm going to let you answer this last question while I answer one from earlier about, how long it takes for a site to get approved and whether families should relocate. I just want to address a question earlier, which was a great question about, in a rat model, if a single dose was very successful. What is the rationale for 5 doses in humans? So again, just to reiterate, so while this is a new drug, this is not a new technology, and Ionis developed nusinersen or Spinraza in spinal muscular atrophy, which has been given to thousands of patients. I don't know all the numbers off the top of my head. Again, this is a drug that cannot be sustained in the system. So this is not a gene therapy that fixes the problem. This just blocks the body from making the protein, essentially. And so there is no expectation that one dose would reverse all the symptoms in a human, and that to knock down all of the protein, you would need to continue to give this drug. So it's a great question. I mean, we're hoping for such successes as seen in the rat models, but that's the purpose of the clinical trial, is to really learn how much GFAP is knocked down from dose to dose, and then how much of that can be sustained over time. In terms of, I'll let Dr. Smith answer this question about relocating and stuff. What I will say is this. If somebody is deciding to relocate within the US, or between countries, I would encourage you to make sure that you had some sort of medical insurance that covers you in a different state or a different country. So first of all, there's those types of issues, right? So even in the US, if someone moves from one state and has maybe, for example, Medicaid and moves to another state, you have to make sure that you have insurance in case you have a medical problem. Now, the trial is paid for by Ionis, but if, heaven forbid, and I'll just use COVID as an example, someone gets COVID or ends up in the hospital because they got pneumonia, those things are not covered by the trial. And so for international patients or someone moving from one country to another, that becomes even more complicated. So there's a lot of planning that would need to go on if someone's going to relocate their family, whether it's within the US or within countries. I also want you to keep in mind that this is several years, right? So, would have to sort of make that decision for your family that you would relocate for 3 or plus years. It's a long time to think about moving to another state or to another country. Dr. Smith, do you have any other advice in terms of releasing information?
No, I think that's all excellent advice. To the question of would it be possible that by the time a site got up, the trial were already enrolled? I can tell you, we're not going to go through 10 or 18 months of effort to get a site up and not have that site enroll patients, right? So we're gauging this as we look at enrollment in the early sites, and we're only going to go to that effort and get that. I mean, these sites are excited to do the trial. We certainly are not going to bring them up and say, sorry, too late. So I wouldn't worry about that particular scenario. As to relocation, I don't know. I don't feel like I can weigh in on that. That's such an individual personal decision. It's a huge life decision. Talk to Amy, talk to the clinician where you're thinking about going to make sure you really understand the impact.
Yeah, and there's these questions. There's a couple of questions. Let's finish that one, and then we'll come back to some of the ones about the baclofen pump and the spinal fusion surgery. So, in terms of people gauging this, and will we be able to tell how many positions are filled? Or there's a question, how many are enrolled so far? As part of the clinical trial process is one where the goal is to minimize bias. And we've actually had some deep conversations about this, Ionis and myself, about, well, can we tell people when this cohort has been enrolled, or this cohort's been enrolled, or this site is quote unquote full? I mean, there's no strict criteria on that. It does create a little bit of a challenge in terms of adding some additional bias into the clinical trial. I know it doesn't seem like that, but if we start getting to the point where we're saying, okay, this is full, and now this is full, or this is full, it actually can introduce bias. So the plan right now is to share when the trial is fully enrolled, but not necessarily update along the way and say there's 5 spots left, there's 1 spot left. I think that can create a little bit of chaos. So I would say if you're interested, contact a site. There are only 42 spots. There is the ability to go up to 58 if it's needed. So for example, if we if we were only able to enroll patients that are, over the age of 50, and we realized that we didn't enroll any children, which is, not going to happen, then we could use some of those additional spots if needed. But the goal right now is 42 patients to answer the question. And back several years ago in Wisconsin, this was very striking for me. Like Anne said, the goal of a clinical trial is actually to show that a drug is safe and effective in as few patients as possible. So I think it would benefit everyone if we could do this in as few patients as possible to show that the drug is both safe and effective. There's some questions in terms of the spinal fusion and baclofen pump. So a spinal fusion surgery doesn't necessarily make you ineligible. It could make it a more complicated lumbar puncture, and the lumbar punctures in this study are not being done through interventional radiology. So what I would say, again, part of the screening process, if someone has had a spinal fusion, I would add an extra step to the screening process where they would meet with the person doing the spinal tap so that they can actually look at the person and their positioning and figure out whether or not they would be a good candidate for repeated lumbar punctures, right? So the worst case scenario is that you enroll someone into the trial and then you can't administer the drug. We don't want that to happen. Or the first LP is successful and then we can't get an LP because of scar tissue and everything else, for all of the other injections, and then the patient wouldn't be able to continue. So it's not necessarily an exclusion criteria, the spinal fusion, but it does add a layer of complexity that will need to be addressed before the patient can be considered eligible. Baclofen pumps, I'd have to, Anne, do you know if someone with a baclofen pump, I don't think that they are eligible, but I'm not sure that I don't think we've talked about that.
Yeah, I think that's talk to the clinician at the site that you'd like to go to. That's going to be a patient specific situation.
Yeah.
Okay, I think we only have 3 minutes left, and there's one question I should answer. Somebody asked whether our research on the rats has shown any negative side effects from the antisense treatment. And the short answer to that is no. You can always say we didn't really look for everything we could have possibly looked for. We're continuing to try to look for side effects, but we haven't seen any negative effects yet in the rat.
All right, while we're watching for some last minute things, I think that my closing remarks would be to please be in touch, with the different investigators and the different sites, share your interest, let people know that you're out there and that you're interested, but please also understand that the process is slow. It's sort of a one at a time and paced over time. So please be in touch. We will share when we can. It's not always possible for us to update the numbers and update everything along the way. But as soon as there's information about children or adults at a site or a country is active, that would be shared more broadly. And Dr. Messing has done a great job trying to share information when the site becomes active or when there's updates to clinicaltrials.gov. And if you have questions, we're here to answer them.
Yeah, thank you everyone for your engagement, for your interest, for your encouragement, for showing up here on Saturday. And yeah, you know how to find us if you have questions.
Okay, well, if there are no further questions, I just want to thank our speakers. Those were excellent presentations. You got a lot of information in a short amount of time. Thank you. I want to thank our sponsors and our audience for making the virtual family conference this year a possibility. And we really hope to see you all in person next year. And also, the ULF Service and Legacy Awards will be presented over the meal break. So if you want to tune in to honor our recipients this year, that'd be amazing. Thank you all.
Thanks, everybody.
Bye.